So we're going to talk about how glucose is used in the body. So first of all, recall that the glucose is directly absorbed in the small intestine. So now it's into the tissue. And a couple different things happens. When that glucose is going to be stored, there's going to be what's called glycogenesis. And so you should know what these various words mean, but again, you don't have to know the structures or all of the individual chemicals. But the glycogenesis, if we look at the first part of the word, it looks like glycogen. That's the large storage molecule of glucose. So in this case, there's going to be glycogen that's synthesized from glucose, and that's called anabolism. And that glycogen now is stored in the liver and the skeletal muscle. So when it's needed, the opposite happens. And that opposite pathway is called glycogenolysis. And so begins with glycogen, but it breaks bonds. So in this case, the glycogen is broken down into the glucose. So if you think about when this would happen in the body, it would occur when the blood glucose is low. So the blood glucose needs to go up. And to do that, all the storage molecules are broken down into the individual units called the glucose molecules, the monomers. There's also another pathway called gluconeogenesis. And in this case, there is the formation of new glucose molecules from non-carbohydrate sources. So let's say that all the glucose stores are going to be used in the body. They're already gone. They're depleted. So now what's left is lipids or proteins. So those lipids or proteins, for example, glycerol and amino acid, those are the building blocks for fats, lipids, and also amino acids. They can be converted into glucose. And this protects against hypoglycemia, because certain organs in the body, specifically the brain, needs a constant amount of glucose. So to summarize the carbohydrate reactions, there's really 4 of them. The first one is glycolysis. And in glycolysis, this is the first part of the cellular respiration. In this case, glucose is converted into pyruvic acid. So again, you need to know the beginning and the end of this pathway. Then there is glycogenesis, and that's the formation of glycogen from glucose. So this occurs when, right after we've had a large meal, that glucose needs to be stored somewhere, especially if there's extra amounts, and it's stored by forming glycogen. Then we have the opposite of glycogenesis called glycogenolysis. And in this case, the glycogen is converted into glucose monomers. And then the last one, gluconeogenesis, is glucose from non-carbohydrate precursors. So the glycerol for fats or the amino acids for the proteins. So really, these bottom 2 are very similar. And then glycogenesis is going to be the opposite of that. So now for the next major type of molecule that's used in our body would be the fats, the lipids. And these lipids are important because they provide a greater amount of energy from the glucose. They happen to yield 9 kilocalories per gram, which is a little over twice the amount of carbohydrates or proteins. So they provide a lot of energy. However, the problem is that it's difficult to break the bonds between lipid molecules, so between the monomers. So what happens is there are lipases, for example, from the pancreas, but there's also bile that helps. So most products of fat digestion, the ones from the diet, are transported and they're stored in adipose tissue. The products of fat digestion, they're transported in lymph. Remember, there are lacteals, which are found in the small intestine, and they're transported in what are called chylomicrons, little droplets. So the chemical digestion of lipids, the result is the monomers. And the monomers for lipids are the monoglycerides and the fatty acids. So the two different pathways for lipids, first of all, the anabolic pathway, when there is larger triglycerides that are formed, is called lipogenesis. That's triglyceride synthesis. And this is when it's going to be stored. Also, glucose can be converted into fat, which that's one of the reasons that high starch diets can lead to extra fat deposits throughout our body. The opposite of lipogenesis then is called lipolysis. So lysis means to cut. So if you can remember that suffix, that will help you to remember that. So it's the breakdown of stored fats into the monomers, which would be the glycerol and the fatty acids. And again, remember, it's the opposite of lipogenesis, because fatty acids then are usually preferred by some organs, like the liver, the cardiac muscle, and resting skeletal muscle. So this diagram shows us the lipid metabolism, and what we see here first is the lipogenesis, where the monomers, specifically glycerol, is then going to be converted into glucose for glycolysis. And so that's lipolysis. The opposite of that would be lipogenesis, the formation of triglycerides. So kind of going in the opposite direction, combining these two. Then there is a pathway called beta oxidation, and beta oxidation is going to convert fatty acids into the chemical acetyl-CoA, which, remember, is the beginning of the citric acid cycle or the Krebs cycle. So the really cool thing about metabolism is all of the different substrates can be used to eventually make energy, which is obviously the ultimate goal. So the quick summary of lipid reactions are shown on this screen. Again, the most important thing really is what's at the beginning of the metabolic pathway and what's at the end. So beta oxidation is a conversion pathway, fatty acids to acetyl-CoA. Lipolysis is to cut, so it brings down lipids to fatty acids and glycerol. So large to small, that would be an example of catabolism. And the opposite of that is lipogenesis. So the formation of larger lipids from smaller molecules. So in order for our body to synthesize structural materials, the lipids play a very important role in the structural materials. If we think of examples like the phospholipids, that would be in the cell membranes and myelin. Also, cholesterol is extremely important. And we now know that it's in cell membranes, but also steroid hormones like estrogen, testosterone, progesterone. And also, it's used in the liver. It synthesizes lipoproteins for cholesterol and fats. These would be things like LDL that you hear about, that's the bad cholesterol. Also, it synthesizes cholesterol from the acetyl-CoA, and to form bile salts, which are needed in the gallbladder. So for protein metabolism, we have absorbable proteins in the form of those amino acids, the monomers, which can be absorbed. And those are very important for these various examples here. So fibrin, fibrinogen, myelin, the insulation surrounding the axons, so to allow that action potential to travel faster. Immunoglobulins, like the gamma globulins, that would be like IgG, for example. Hemoglobin and myoglobin, so to transport the oxygen in the blood cell. or in the muscle, and then also the actin and the myosin used for contraction within the sarcomere. So the proteins, they're not stored in the body. They're going to be synthesized from what we eat. And when they're in excess, they're oxidized for energy or converted to fat for storage. So it's kind of like they're stored but in another format. So they're converted into the fats before they actually can be stored.